Hi, Ben.
Hi, Marcel.
Good luck.
Thank you.
Hello, Mr. Candidate.
Hello, highly esteemed Professor Marsh. How are you doing?
I'm good, thank you.
Oh, good. You guys all look so sharp.
Yeah, so we're going to start. So thank you all for coming, ladies and gentlemen, and Mr. Candidate, Matern. I am opening this online academic ceremony in which you, Mr. Benedict Mark Matern, will defend your academic thesis prospecting the MHC, a bioinformatic view of HLA polymorphism and gene organization. And before we start the defense, I would like to invite you to present a summary of your study and the conclusions of your thesis in the forthcoming 15 minutes. You can start.
Thank you to the Prorector for giving me this opportunity to present the results of my PhD studies. Thank you to the esteemed members of the Corona, and thanks to my family in Minnesota and my friends all over the world who can join as well. So I'll share my slides here. Here it is. So although a traditional defense ceremony was not possible, I am a bioinformatician, and that training helps me to use technology to share these scientific ideas with friends and collaborators. So this is my Lekenpraatje. That's a Dutch word for the layperson's talk. So this presentation is intended for the general audience, but I've also included some points for the experts as well. My thesis is called Prospecting the MHC. Prospecting is a theme that developed early in my studies. A prospector is a person who explores a region, who's making maps, and who's searching for gold. In my thesis, I'm using bioinformatics to make a map of the patterns of the MHC, to search for the gold that's hidden there, and to come up with ideas about the results of these patterns. So this thesis is focused on bioinformatics, which can be a bit complicated to define. The original definition came from Utrecht University about 50 years ago, and this definition has evolved over time to include more modern databases, algorithms, software, and analysis techniques. So I've boiled it down to my definition, the study of information in biology. Bioinformatics is important for two main reasons, which correspond to the two sections of my thesis. The supportive role of bioinformatics is covered in chapters 2 through 5, where I describe tools and techniques that can be applied to a variety of research projects. More importantly, bioinformatics allows the development of scientific ideas, which is discussed in chapters 6 through 9. Although bioinformatics contributes to many important areas of study, my thesis is focused on the study of the HLA genes in the white blood cells. So your blood is filled with many different types of cells. In these images, you can see red blood cells and white blood cells. Red blood cells are defined by blood groups. A person might have the A, B, or O blood type. White blood cells are defined by HLA. HLA stands for human leukocyte antigen because these are on leukocytes, which is another name for white blood cells. The markers for HLA are similar but more complicated than the ABO blood groups. Instead of just 3 different variants, there are many, many different variants of HLA. These HLAs are defined by genes, and the blueprint for these HLA genes is found in your DNA. So you know that DNA is the blueprint for the machinery in the human body. This means that all of your proteins are encoded in the DNA somewhere in your genome. Humans normally have 23 chromosome pairs, and these are pairs because you get 1 copy from your mother and 1 copy from your father. Each of these chromosomes have different genes on them, and these genes might be related to eye color or some other function. If we zoom in on chromosome 6, we find a special region called the MHC, the major histocompatibility complex. The MHC is also known as the HLA region because if we zoom in further, we can see that there are many HLA genes located here. These genes are divided into different classes, which have somewhat different functions. So what is the function of HLA? HLA is a protein that is embedded on the outside of white blood cells, kind of like hair is embedded on your head. The shape of the HLA is the important part. They have this peptide binding groove at the top, which is shaped like a hand. And this hand can find a bit of a nearby protein and hold it up in the air saying, look what I found. That way your immune system can respond. It can take a look at it, decide if it's friendly or dangerous. So this function of HLA is called antigen presentation. An important concept for HLA is that it's polymorphic. That means there's not just one HLA, but there's many different HLAs. It's so polymorphic that people have to give special names to these different varieties of HLA. These names tell us a little more about the shape of the HLA protein and the DNA that encodes it. And by using these names, it's easier for us to understand each other when we talk about HLA. So why is HLA so polymorphic? It's because we need to fight off a wide variety of foreign compounds. Maybe you've heard of this COVID-19 coronavirus. This is a nasty virus that can get inside your cells, and it starts to make these virus proteins that can cause illness. An HLA can hold these coronavirus proteins up in the air, and hopefully your immune system will recognize it as dangerous and destroy the virus. But COVID-19 is only one disease. There are lots of different HLAs for lots of different foreign compounds. So in this way, HLAs are very good at protecting you, but in the case of transplantation, they might actually cause problems. When you're bringing together cells from two different people, you're introducing foreign compounds. So that's why in transplantation, we often want to match the HLA genes of the patient and the donor. If we want to match the HLA, one of the best ways is to match the DNA that encodes the HLA genes. If we compare the DNA sequence of the patient with the DNA sequence of the donor, we can see if they're a suitable match, and this can improve outcomes in transplantations. The technology for sequencing DNA is constantly improving. I am most interested in this MinION sequencing device. With MinION, we can sequence multiple HLA genes together. Sequencing multiple HLA genes together allows us to define HLA haplotypes. Haplotype is just all of your HLA genes together on one continuous stretch of DNA. If we can figure out the arrangement of HLA genes on a haplotype, we can improve outcomes of transplantation even further. So we've discussed a very brief background of HLA and how HLA research applies to the clinical setting. I'd like to share with you some of the ways that I have contributed to this field. My thesis is divided into two parts, and part one is called Rules and Tools of NGS Analysis. In this section, I discuss the creation of tools and techniques that provide a supportive role of bioinformatics in HLA studies, and I'll highlight chapter 4 in some detail. Chapter 4 is about the application of MinION sequencing to routine diagnostics. We've already discussed that we can use DNA sequencing to match the HLA between patients and donors. There are many ways to sequence DNA, but some of the older technologies have some limitations. Now we have this MinION. It's portable and fairly cheap. It's smaller than your cell phone, and it has this USB connection, so you can use it on most laptops. The MinION has this sheet of silicon with nanopores embedded into it, which are colored dark blue in the figures here. So nano means small and pore means hole. So these nanopores are just small holes. These nanopores have special proteins attached to them that can push the DNA through the pore. As the DNA passes through the pore, it's creating an electrical signal, and this signal changes depending on which DNA sequence is inside the pore. We can use special computer programs to interpret the electrical signal and determine the sequence of the DNA. And the cool thing about the MinION is that it can sequence very long pieces of DNA, which means it allows us to sequence the entire length of HLA genes. So using the MinION, we developed a new method to sequence HLA for clinical diagnostics. It's fast and easy to use in the lab, and we can use it to type 3 of the HLA genes. That's HLA A, B, and C, which are circled in red in the figure here. We tested this method on 100 samples, meaning we compared the MinION results with results from other sequencing technology. This technique is now available in the Maastricht tissue typing lab to be used for transplantation matching. But in addition to the clinic, it's also a useful tool for research. So with this new technology, we have new tools to answer scientific questions. The second part of my thesis is called What's in a Haplotype, which is focused on some of the scientific questions related to HLA. Specifically, I looked at the polymorphism of the HLA genes, but I also looked at how the HLA genes are organized within the MHC. This figure shows one general arrangement of the HLA genes, but it's actually more complicated than that. There are many different haplotype patterns, and I've helped to define some of these patterns. Again, there's not enough time for all of these chapters, but I will highlight chapter 6 and 8. Chapter 6 is focused on the genes in this HLA-DR region, which I've circled in red here. This chapter is about understanding the polymorphism within my favorite HLA gene, DRA, and determining how DRA relates to the other genes in the region. This helps us understand the nature of HLA-DR haplotype patterns. As a bit of a background, we already knew that DRA lies within several different haplotype arrangements. Different individuals have different DR haplotypes, and some DR genes are only present on certain haplotypes. We also knew that DRA is not as hyperpolymorphic as some of the other HLA genes. Before we started this project, there were only 7 unique DRA sequences in the database. And that's compared with other HLA genes that have several thousand unique sequences. So that means for most people, the DRA protein has basically the same shape. The reasons are complicated and not fully understood, but I did discuss some of these in chapter 6. So we sequenced 100 DRA samples, and we found that DRA is actually more polymorphic than we thought. We identified lots of new sequences, and we submitted them to the database to share with the community. And this polymorphism was rather unexpected. It goes against some of our previous assumptions. So we took this newly identified polymorphism and compared it against the other genes in the HLA-DR region, and we used bioinformatics to determine how DRA relates to the DR haplotype patterns. And what did we find? This chart shows how the DRA gene is related to the DRB1 gene. Each color here represents a different variety of DRA, and each column represents a different variety of DRB1. We found that specific varieties of DRA were linked with different varieties of DRB1. For example, on the far left, DRB101 is always linked to this blue DRA pattern. And on the far right, DRB116 was always linked to this yellow DRA pattern. The other columns are a little more complicated, but we can also find some interesting patterns there. The conclusion from this is that by using bioinformatics, we can take a close look at how these genes are related. And this might reshape some of our previous assumptions, and it might redefine how we describe these genes and haplotypes. And that gives us a better understanding of the haplotype patterns. Chapter 8 is focused on a very special gene in transplantation, that's HLA-DP. So we know that sequencing HLA is important, and we know that determining haplotypes is even more important than the individual genes. And DP haplotypes are a special case for a few reasons. So when I say HLA-DP, I'm talking about 2 genes, that's DPA1 and DPB1, which are circled in red here. These genes are so close to each other that they're actually overlapping. The overlapping part contains this important region called the promoter. In the past, we have looked at these genes individually, but since they're so closely related, it makes sense to think of them as a single combined unit. Now that we have the sequencing technology and the bioinformatics capabilities, we can start to do that. We sequenced 100 samples for the region covering DPA1, the promoter, and DPB1. Since these regions overlap, we can determine the continuous sequence over the whole combined region. We looked most closely at this important promoter region between the 2 genes, and we defined 11 different promoter patterns and gave them the names ProDP1 through ProDP11. We took our panel of complete DP haplotype sequences and grouped them based on the patterns found in the promoter. And that means if we know the sequence of the promoter, we can predict some of the potential shapes of the DPB1 and DPA1 proteins on this DP haplotype. The more we know about the shape of these HLAs, the better we can understand their role in transplantation. So what we're doing here is we're using sequencing technology and bioinformatics to define haplotype patterns within the HLA-DP region. And by defining haplotypes, we increase our scientific understanding of the HLA region, and the more about haplotypes, the better it is for transplantation. So I want to leave you with this takeaway that bioinformatics is taking a more important role in science. As we move into the future, the role of bioinformatics will not decrease, it will only become more important. In many cases, especially in times of social distancing, research projects are moving from the laboratory to the digital space. And as we develop new tools for analysis and use new technology to gather data, a whole new pile of biological data is made available. More bioinformatics expertise is needed to figure out ways to interpret the data, to develop new databases to store the data, and to develop scientific questions. So bioinformatics can change or redirect how we think about a biological problem, and in that way, it improves our ability to ask scientific questions and to find answers to these questions. So thanks for your attention during my presentation. I look forward to fruitful discussion with the members of the Corona, and I'd like to give the word back to the prorector.
Thank you, Mr. Matern. Thank you for your presentation. And we are now going to start the opposition, and the opposition will be opened by Professor Zuurhausen. He's a chair member of the assessment committee of your PhD, and he's also head of the Department of Pathology in our academic hospital, the Maastricht University Medical Center. Professor Zuurhausen.
Thank you. Mr. Candidate, I'd first like to start to express my sincere congratulations to you and your promoter team for your fantastic book. And actually, it's difficult for pathologists already to talk about, or for pathologists to talk about bioinformatics. Bioinformatics and immunology is maybe even more a challenge. So let me start with some simple questions, because you were talking about polymorphisms, and may I turn to your proposition? Start with your proposition number 6. It's so short that I can read it even myself. So it's every SNP is important, you say. What do you mean with this?
So I wanted to include this proposition because I do think it's very, it's a good prompt for discussion, and I think a lot of people would disagree with this. What I found is we've sequenced, for instance, in this DRA project, we found a lot of these interesting new polymorphisms, and a lot of people would think that they're just not functional, they're not expressed on the protein, but it turns out they're actually pretty interesting. So when I say every SNP is important, obviously the easy ones are the exon SNPs that are encoding the proteins, but there's also these non-coding proteins and intron SNPs that should also be considered. Even if they're not, expressed in the protein, it might be a target site for microRNAs, or it might affect expression somehow. And these could also be important. There's also polymorphisms outside the gene that may or may not have an effect on expression, but it's still interesting to look at because they may be linked to functional polymorphism, may be part of patterns across the whole MHC. And even if there's no functional relevance there, it can be important for like evolutionary studies, figuring out how these regions have evolved.
So excuse me if I interrupt you. But then you're talking about immunology, SNPs for immunology.
Yes, SNPs for immunology. Well, I think if you're looking for the SNPs that are important for immunology, then the ones that control the shape of the protein and the ones that control expression are the most important. When I say that every SNP is important, it would be for interesting research objectives.
In immunology, or?
Well, I wouldn't say that every SNP is critical for immunological function. But I would say that it's important and interesting to look at.
So I would wish that you could talk to the reviewers of my papers already 10 years ago, in which we characterized several polymorphisms and SNPs in a gene called kindlin, which were definitely not important, at least according to the reviewers. That's so basically, how do you determine importance?
There's a few different ways to rank SNPs. I'd say that there's major polymorphisms and minor polymorphisms. There's certain specific polymorphisms that would like divide HLA alleles into two distinct clusters, or several distinct clusters. So those ones would probably be ranked as the most important. SNPs, for instance, at the end of a homopolymer, maybe have 27 or 28 A's. It depends on context, but those might be, less important. But in many cases, the difference in a homopolymer could result in a null allele. And so I guess there's a few ways to rank those. It mostly would be based on function, mostly would be based on, the difficulty of sequencing and its relevance in immunology, and maybe slightly below that would be its relevance in evolutionary studies, or its usefulness as a marker for other polymorphism.
Okay, thank you very much. Actually, may I ask you help to read out proposition 7 for us?
Proposition 7, when associating SNPs with disease, it can be hard to tell the spurious from the serious.
May I ask you to explain what you mean with this?
Yes, so we're talking about disease association here. When we're looking especially at the MHC region, we do some GWAS studies, that's genome-wide association studies, and these might be grids of oligonucleotides that are testing lots of polymorphism, maybe just across the MHC or across the whole genome. Since we're dealing with so many of these separate oligos, it can, you're testing lots of different things, and just by the laws of large numbers, you might get some spurious associations with some polymorphism with the disease. So when you're looking at the results of these studies, you got to be careful to determine which ones are actually related to a disease and which ones are just a spurious statistical result.
Okay. Well, actually, I would like to take your or draw your attention to page 70 and 71, in which I am coming from the field of oncology, actually from virus-associated oncology.
Which page was this?
70. I think it's in 71 and 70. Yeah. I see those phylogenetic trees.
Yes.
Based on different polymorphisms. So basically the distance of the trees, of within the tree, indicates how far, in as much these are related to each other. This is by sequence, isn't it?
Yes.
How would those phylogenetic trees look like if you would try, or maybe you can answer whether this is possible at all. How would those phylogenetic trees look like if you would cluster them according to function?
Clustering them according to function. Well, thank you for your follow-up question. I think if we were going to cluster these alleles based on function, the first place that I would look is their relevance in immunology. Since these are HLAs, it would be the antigen presentation region, which for class 1 alleles is exons 2 and 3. So if I wanted to cluster these alleles based on their function, I would want to cluster them based on the polymorphism just within exons 2 and 3.
Okay.
So it could be interesting then to take these trees and recreate them focused only on the antigen presentation domain and see how they look in that context. These trees were drawn, I believe, with full-length sequences that were aligned with each other.
Okay. Basically, do you think it would make sense to do this?
I do think that it would make sense to do this. It's we are just drawing these trees based on how the sequences are different from each other, and looking them in context of how they're functional is very good. It might give us clues for a way to match these alleles rather than just their allele names. If we could divide them into clusters, maybe sequences that are in a specific cluster might be better for matching.
Okay, so I think I have time for one last question, actually, and this is, I have heard someone saying, I think it was the great Professor Tilanus himself, when he said that if nanopore sequencing is able to sequence, or if you're able to sequence with this technology, the HLA gene, then it's basically could sequence everything. So would you agree on this?
So if I understand your statement, it's if you can sequence the HLA, then you can sequence everything. And this, when I was choosing my propositions, I used to have one that was like, DNA is simple unless you're looking at the MHC. And I think what we're getting at here is that the MHC is a very complicated region. And if we can use this MinION then to sequence these HLA genes, then it should be possible for basically any DNA. And I think if we can handle these complex, these hyperpolymorphic regions that have these strange repeat regions and things that are difficult to handle, then we should be able to sequence the rest of the genome.
So, of course, asking for the background for pathology, but I see that my time is over, and I think that Professor Ernst Jan Spee might ask you some additional questions about this technology. So thank you very much for answering my questions. I'm very happy with your answers. So I return the words back to the prorector.
Thank you.
Thank you, Professor Suhausen. The opposition will be continued by Dr. Spearings, who's a member of the assessment committee of this thesis, and he's associate professor and director of the HLA tissue typing laboratory in the University Medical Center in Maastricht. Dr. Spearings.
Thank you, Mr. Prorector. Dear Mr. Candidate, before I start the discussion with you, I would like to congratulate you sincerely with your thesis and the results that you presented therein. And I certainly want to extend my congratulations to your supervisor and co-supervisor. The end result, I think, shows a nice example of a multidisciplinary approach in the field of histocompatibility, covering both technical methodologies regarding HLA typing and bioinformatics, and scientific approaches, all used to gradually move from the immunogenetics towards the immunogenicity and antigenicity. And being a transplantation immunology, I can really appreciate this approach, particularly because it shows that in the end, immunology is about the phenotype and not the genotype. Mm. As such, it's an honor for me to be allowed to take part in this thesis defense ceremony and discuss with you some aspects of your thesis. So having said that, it won't surprise you that despite my appreciation for the bioinformatics and the immunogenetics, my focus will be on the chapters 7 and 9. And in the context of these two chapters, I have two issues that I would like to discuss with you and hear your opinion and view about. Mm-hmm. And for that, I would like to take you to chapter 7, page 164. Maybe I share that with you as well. And I would like to start with a general question to hear what your opinion is on one of the statements that you made in the introduction. Because what you say, and I quote from page 165 in the first paragraph, in standard HLA nomenclature, HLA alleles are given specific and meaningful names. And you refer here to an important paper of the esteemed Professor Marsh, member of this assessment committee. But to avoid any confusion, the reference is linked to the first part of the sentence and not the second part. My question, however, relates to the latter. So I would like to hear from you how meaningful the HLA names are, in your opinion, also taking into account the observation that you described in chapter 7 and chapter 9.
Thank you for your kind words and your very interesting question, highly esteemed opponent. So if I understand your question, then you're asking about the nature of the HLA nomenclature and that they're given meaningful names that describe their functionality and how that applies. I do think that the HLA nomenclature is a very good system for understanding and communicating how these HLA genes are functional. It gives us clues about, first, in some cases, their serology, some cases, their peptide sequence, and then some of the non-coding polymorphism that is around it. So it does give us a very, at least clues towards the functionality of this. And if I am correct, chapter 7 and 9 are the B15 alleles, and the chapter 9.
Yes, chapter 9 is the B15 and chapter 7 is the DRB113 haplotypes
Yes. Yes.
and the immunogenicity thereof.
Yes, thank you for that. So regarding chapter 7, the DRB113 alleles, that's a very interesting case because there's a lot of these different genes here that have very similar names. These are all DRB113 genes, which are, clustered together in this in the standard nomenclature. So at first glance, they do look like they would be very similar. Based on the results of the DRB113, I would say that in many cases, the nomenclature may not fully communicate the full distinctions between some of these alleles. In that chapter, we found that these different genes had kind of a different context on there. The DRB113s were linked to different patterns in the DRA, and in that sense, they live on distinct haplotype patterns. So that could be a consideration for when we're naming these alleles, or at least defining how alleles live on the haplotypes. So in that context, the nomenclature is pretty good, but as you suggest, there could be improvements if we're thinking about it in a haplotype context. Regarding B15 alleles, in that chapter, we were defining, or at least predicting, some serological types based on the amino acid patterns. These B15 alleles do have some distinct serological types, and for many of the sequences, we just do not know the distinctions between some of them, and with some unknown sequences, we might not know the serological types. So we can define them as B15, which gives some idea about the polymorphism, but it may not give a full idea about the function there. So we can define which alleles fit into groups, but dividing them based on serology is a little bit more difficult. And I guess in that way, it's important to do more follow-up studies to determine the immunological behavior and see what ways we should be naming them, I guess, in that way.
So if Professor Marsh would give you the freedom to set up a new nomenclature from scratch, what fundamental changes would you make to this nomenclature based upon your experiences?
Well, thank you for the follow-up question. I think if I was to define a new nomenclature, I think the nomenclature we have is very good, and it's a good start. I think it's a very good approach, and I know Professor Marsh feels the same way. If I wanted to design a new one, I would want to include the idea of haplotypes. This nomenclature doesn't really consider, its context in the MHC, so I think it'd be interesting to look at ways that we can include, entire haplotypes in there. I'm imagining, we have a bunch of reference MHC sequences with just like annotations for different HLA alleles, and the annotations would include, the normal nomenclature that we have now for exon and intron patterns, but maybe it can include like more of the context of where it is and what genes are around it. And I don't have an answer for exactly how to do that, but that's an interesting place to discuss that.
Okay, thank you for that. And then I would like to move on to my second question, for which I would like to go to pages 170 and 171, where you mentioned the ignorance of the DR13 mismatches by the immune system. So the DR13 alleles are associated with protection for autoimmune diseases and recurrent miscarriages, and you mentioned that the mechanism underlying this ignorance may either be due to specific regulation, thymic deletion, or to absence of serological determinants. Mm-hmm. Demonstrating specific regulation and thymic deletion in these situations may be complex and take quite some laboratory work, although not impossible, but I'm confident that you have all capabilities to address the absence of serological determinants. So I challenge you here to use your expertise and to elaborate a little bit more for us what experiments could be able to reject or confirm these latter hypotheses using your bioinformatics skills.
Short answer, please.
Yes, so you're suggesting that DR13 has a reduced immunological significance and what I could do to test this.
Right.
I would like to take some of these sequences and determine what epitopes are present for T cells and for B cells, and see, maybe in some ways we could predict how they would react with T cells and B cells. And I know that there's a lot of biological or bioinformatic tools that are available for doing that we're both well familiar with. So I would start by using that to determine what the T cells and B cell epitopes of these might be.
Okay, thank you very much for your answers, and I would like to return the word to the prorector.
Thank you.
Thank you, Dr. Spearings. The next opponent is Professor Marsh. He's a member of the assessment committee, and he's director of bioinformatics and the deputy director of research in the Anthony Nolan Research Institute, London, UK. And thank you for being online rather than coming to the Netherlands, Professor Marsh. But our university appreciates the guests from abroad always very much. So thank you again. The floor is yours.
Thank you, Mr. Prorector. Thank you, Mr. Candidate. I thoroughly enjoyed reading your thesis. Congratulations to you and to your supervisor and co-supervisor. It's really nice as somebody who personally started off working in a laboratory and became a director of bioinformatics, whatever that means, to read a whole thesis that is directed at bioinformatics. In your thesis, you have chapters which deal with sequencing data that's derived both from Sanger sequencing and standard sequencing, and both then next generation and third generation sequencing. Would you just like to briefly highlight the differences between the next generation and third generation sequencing that you've looked at data from, and what the benefits are of those?
Well, I want to thank you for your kind question, esteemed opponent, and for your kind words about my thesis in committee. So you're asking about the differences between the different evolutions in sequencing technology and as they move forward and what improvements are made. Specifically between NGS and third generation sequencing. I think the biggest benefit of so NGS technologies are usually based on shorter reads. We have regions of, I think, around maybe 200 bases or so that can be sequenced at a time, and piecing these together is a task. You need some bioinformatics to take these shorter pieces of DNA, align them together, and understand the continuous sequence. That would have the weakness that if you have two polymorphisms that are completely separate from each other, it can be difficult to understand which HLA sequence the two different polymorphisms belong to. So I would say the benefit of moving into third generation sequence is that we can sequence longer stretches of DNA at one time. The two main technologies here are MinION or nanopore sequencing and this PacBio sequencing, and both of those would allow you to sequence the entire region at one time. That makes it easier to understand which HLA sequence a polymorphism belongs to, and in that way it makes analysis and understanding the HLA sequence a little easier.
Excellent. Okay. Moving on from that, the Oxford Nanopore's MinION technology is let's say relatively new. I'm being kind there, and users have had variable experience with it. I know in my own lab we've looked at their technology. And while it's clearly you're able to get lots of nucleotide sequence data from Oxford Nanopore, do you really consider, or can you back up why you think, I suppose, this data is sufficiently accurate for doing HLA typing? And if so, what's your role in achieving that success on that MinION technology informatically?
Yes, and this is the difficulties with using MinION are brought up quite frequently, and this is indeed a challenge. When I began my PhD in Maastricht, I started writing some software that would handle this data. And the first thing you do is take a reference sequence and align some data against a reference sequence and just see what it looks like. And immediately, you can see that it has some challenges understanding these homopolymer regions and, getting completely accurate results. I first started with my analysis by doing some de novo assembly, taking these reads and assembling them to each other. and getting a sequence based on that. The challenge there is that you have to assume the data is correct and move forward based on that. What I found is that it's easiest to understand the data if you categorize where the difficulties are and try to respond to them. So in many of the HLA sequences, we know where the challenging regions might be, specific homopolymers that we need to watch out for. We need to understand the data that you're looking at and if it can be applied and to get a meaningful answer, and have a backup plan in that case. I know that the Maastricht lab recognizes these difficulties with MinION, and while it is a great tool that's convenient and easy to use and gets data, if we see a troublesome spot where there might be difficulties, then we do need to follow it up with a secondary method of testing. So.
Much of the work that you have presented lies around phasing, polymorphisms, and understanding haplotypes.
Mm-hmm.
And you talk about that in terms of HLA DRA, and I'd love to spend a lot of time, and we've already talked about DRA quite a lot, and it's been mentioned before today, so I'm not going to delve into DRA. But in terms of using that information in terms of haplotypes and the work you present in your chapter on the DR13 families and assigning haplotypes, is that, or can you see a pathway for the information that you are generating here moving back into a clinical lab? And do you see a role in using this haplotype information in matching donors and recipients in hematopoietic stem cell transplantation? Is that something that's already happening? What is that pathway?
Yes, that's a very good question. So the technologies and the techniques for matching for transplantation just seem to keep evolving from a small region based on exons into a full gene or multiple genes. And a paper came from your lab that was about using full-length high-resolution sequencing is going to improve results even more. And from like Petersdorf studies, we see that including the haplotypes is improving things even further. So I would just say the more information we have, the better it can be applied to transplantation matching. Can we use it directly now? I think we need to, get the ball rolling on actually acquiring this data and, having it available for use in transplantation. We're currently matching based on just a few genes, but if we do have, a small donor pool, we could start to test these haplotypes. Consider it in matching, or at least have the data available so we can, we can prove that this will work in transplantation settings.
And then sort of finally on that topic, how would you go about showing that matching at the level of haplotype matching, let's say for the DR13s, is going to have an improvement effect on the outcome of HSCT?
Short answer, please.
Yes, I would start with some retrospective studies. We do need to gather some haplotype information. We do need to probably do some matching based on the the DRB as we're doing currently, and then take a look and see if these haplotypes are going to be useful in matching. So gather the data and then take a look at it using techniques we have.
Thank you. Thank you for your answers, Mr. Candidate, and I hand the word back to Mr. Prorector. Thank you.
Thank you, Professor Marsh. Then the next opponent is Professor Speel. He's professor in clinical molecular biology in our institute, and today also the secretary of this committee. Professor Speel.
Thank you very much, dear Prorector. Mr. Candidate, first of all, I would like to congratulate you with your nice PhD thesis, which I have read and which is quite some extensive work to read up. I'm not a bioinformatic person, but I like to try to connect data, biological data, with bioinformatics. And so it's very interesting, I think, to think about. And I, of course, also would like to congratulate the supervisors and family and friends from your side. Yeah, with respect to proposition, my colleague Axel Truhausen that every SMP is important is very philosophical, maybe kind of proposition. And I liked very much proposition number 10, DNA is like a Taco Bell. I've been in in Canada, Vancouver Island, and I've joined there very much the Tacofino bus. And maybe you could answer me shortly which 4 ingredients you exactly meaning these nice tacos.
Well, thank you for your kind and thoughtful question about Taco Bell, highly esteemed opponent. I know my brothers are watching back home, and they'll agree that sometimes you sit in the Taco Bell drive-through and you can not really decide about which thing to order, if you're going to get a burrito or a taco or a chalupa or whatever. So we have our tortilla, our meat, our beans, and our veggies, and those are the core ingredients of basically every menu item.
Okay.
In Taco Bell.
And so going into the direction of the DNA, then of course, DNA basis, you mean?
Yes.
With respect to your, I would like to go to chapter 4, and actually what you are doing there is trying to get the nanopore sequencing to try to let it become the standard or a new standard for sequencing. And so you compared it with the gold standard Sanger sequencing. And so one of my first questions would be, if I look at that chapter, would be, when do you think you can consider nanopore sequencing to be the gold standard? What is actually the things you have to do? Do you think that you are there with your validation study, or what is, how do you look at that?
Yes. So in order to be considered the gold standard, it has to be, accurate and reliable, and it has to be able to sequence full length and get accurate, usable results every single time. The current benefit of MinION is it does allow us to look at full length sequences with, the difficulties that I discussed with Professor Marsh, that there are some considerations when you're using that data. The difficulty with this data comes mostly from the base calling models. This is the software that's converting the electrical signal back into the DNA sequence. There's a lot of work that's continuously happening on these. There's always constant improvements and different models that can be made with that. So I'm hoping that at some point these, the computer systems and the analysis techniques that we use will be able to get a 99.9% accurate read of the full length of the gene each time. So when we get to the point where we have a portable, cheap, and usable system that works every time, that's going to be the point where we can define this as the gold standard, and we're moving in that direction.
Yeah, moving into that direction. What do you exactly mean with that? Because you already need 2 kind of sequencing or analysis systems, and sometimes one is right and sometimes the other is right, if I'm correct. So how should I see that, and how should I translate that if somebody else should use it?
Yes, so using these technologies is pretty context specific. I think we're mostly using this MinION for quick and easy typing that's able to get a good idea about the polymorphism across the region. If we want to specifically look at one SNP and make sure that we have full coverage of that, then or if it's a region in a homopolymer, then perhaps we would need to confirm that with a different technique. So when I say we're moving in that direction, I say that we need to move forward with these technologies and start gathering data and understand the data. We've already been applying it to the setting, so in that way we can prove that it does have its use.
So actually you are not yet close to golden standard. I mean, how about, for instance, one of the things I thought was interlaboratory or reproducibility studies. Have you done something like that, or how is that moving on at this moment?
So with the reproducibility studies, we are validating the sequence to make sure that it is correct. I know that one important concept is this is a technology that's moving forward. We need to continually improve, and I do feel that we are continually improving. If you think about, like, are we defining the gold standard, or getting to the finally to the finish line where we can say that this is the point where we need to stop and we have everything that we need to do. I don't know if we're ever going to get there. I think we need to just keep improving, using the best technology that we have, and like you say, with reproducibility studies, we do need to validate that it works and that the data makes sense and that it's usable in the laboratory. But it's going to continuously improve.
Thank you. Thank you very much. I have to look to the prorector. Do I have a short question or?
Short question, short answer.
Short question, short answer. Well, the short question would be in the light of cancer therapy. There's a lot of have been a lot of things done now on immune checkpoint inhibitor therapy, and I would like to know from you, and maybe you can try to keep it short, is where do you think HLA typing can maybe bring us there? Or do you have a short idea of how I could predict maybe if such a therapy would be working or not? Do you think that there are possibilities by using this as a new HLA typing technology?
Well, thanks for your question. So you're talking about checkpoint inhibitor proteins, and I think that these are treatments for cancer where we could
cancer
therapy. I think we'd have to discuss that in some detail. I do know that there are plenty of targets within the tumors that you can look at using sequencing. If you look at you can identify single tumor cells and kind of do some sequencing based.
So I mean, particularly in the HLA context.
Mm-hmm. Well, I do. So if you're looking at tumors, it could be that HLA is underexpressed or overexpressed. I think it could be that HLA is silenced, and in that case, you could take a look at what RNA is actually being expressed in the tumor for the HLA, and maybe that would give you an idea about how this tumor is operating.
Okay, thank you very much for your answer. I would like to give the word back to the prorector, and thanks again.
Thank you, Professor Speel. Then we're going to the final opponent, and that is Dr. Meeks, who's a clinical chemist in our Maastricht University Medical Center. Dr. Meeks, the floor is yours.
Well, thank you very much. Also from me, Mr. Candidate, my congratulations with very nice thesis. Of course, I work in a lab next to yours, and when I was invited to participate in this defense, I thought, how hard could it be? A lab is a lab, but I must admit, it took me quite some effort to go through your thesis, but I think it's very well done, but it stretched my personal boundaries to understand something that is not directly in the topic that I work with every day. Despite that, you did a very great job, and I want to congratulate you and your thesis team with this performance. My first question would be related to chapter 6, and what surprised me there on chapter 6 is maybe that you sequenced 98 patients for full-length HLA DRA, and you say in your abstract that you found 20 novel SNPs that with intronic sequences that were not available at that time in public databases like the IPD, IMGT, HLA. And while reading that chapter, I thought it maybe was some kind of a missed opportunity to really show what bioinformatics actually can do, because maybe correct me if I'm wrong, but if I understand it correctly, it showed actually that the polymorphisms that you identified with your sequencing technique were maybe not already known in the IPD, IPD IMGT HLA database, but they were known in the 1000 Genomes database. So also the subsequent haplotype reconstruction that you actually conducted and that you present, to what extent would that be actually possible with doing only in silico work? So not sequencing at all and just trying to employ your bioinformatics skills and align or combine these two existing databases and actually come to the same conclusions?
Thank you for the interesting question, esteemed opponent. So it looks, if I understand your question, we're talking about the polymorphism in DRA and how we could just do studies like this in silico without actually sequencing some of these samples. When I'm talking about the sequences are missing in IPD IMGT HLA, that is, the current best database we have for HLA sequences. Those are the ones that are curated, and there's a close look that's taken at them, and they have, these meaningful names that we've been discussing. So I think that we want to have it in this high quality database there.
Mm-hmm.
When you do look at the 1000 Genomes database, you are correct when you say that many of the polymorphisms we identified are already known in this database there.
But it was
a bit of the weakness.
All of them, right, or not? All polymorphisms you identified were there, right?
I believe all of the, all within the gene, all of them were there. I think outside the gene, there's a few.
But what surprises me then for you, bioinformatician, if I may interrupt you briefly, but because what would make more sense to me is then first employ your skills and go through all the available databases, see what you got, then actually fill up the gaps that you, or define what exactly you would like to do, and then sequence specifically towards what you would be looking for. And this seems like another approach or another direction, and it almost seems like a missed opportunity, like having a chapter focused on a technique rather than which is of course nice, and it's the minion sequencing is without doubt is an improvement to what you have. But I think what's even nicer is that you actually can use your skills as a bioinformatician, and that is when data becomes really valuable, is by combining it from existing sources and then tell the people in the outside world, look what we found and what is there, it was there, but it was hidden basically, and you found it. Is that a misconception that I have, or is that, why this choice?
Mm-hmm. Well, I would say that it was kind of a two-pronged approach. We were doing this amplicon-based sequencing and also the analysis of the existing database, so we can kind of combine these two approaches and understand the polymorphism in that context. The one weakness with just using the 1000 Genomes database and combining that to understand that is we don't really know much about the linkage disequilibrium between many of these SNPs on there. They're just reported with like a frequency and what sample it came from, if it's from like a messenger RNA or where it came from, and it's hard to tie them together into complete HLA sequences.
So if I understand correctly, there's no granular information about what the exact linkage disequilibrium between the SNPs is in those databases. It's not on that level.
I think there may be some information available, but it's not on the level that we need to understand the full length of HLA sequences. The 1000 Genomes is a genome-based project, so there are some limitations within the MHC region. They do have some really interesting data, and it's especially useful for looking at some of the disease associations that have been found, but looking at the linkage disequilibrium between genes and SNPs is a little more difficult.
I see, understand. Well, my next question would be on something that you mentioned in the discussion on page 223, and you discussed quite extensively the definition of the borders of a gene, including the 3' and 5' untranslated regions. And you also try to define, if I understand correctly, the number of kilobases that you can assume to be that actually limits or borders the gene. And I would have two questions about that. Is that important from a transplantation perspective, or is that more that you elaborate on that? Is that more from a general scientific view, because that I would understand. But from a transplantation perspective, if I would look at it, I think the only thing that probably matters is that you have that you know where there is possibly an incompatibility between the donor and the accepting patient, and whether that is the true culprit variant or some variant in linkage equilibrium doesn't really matter. Or well, how should I see that?
So you're talking about the borders of a gene and how far is necessary to define this gene and whether it's relevant in a transplantation setting. The first thing that comes to mind in the DPB1, there's a SNP that really, I forget the number, it's really far downstream from the 3' region of DPB1. This is something that's been demonstrated in some Petersdorf papers about this is controlling or at least related to the expression levels of some of these DPB1s. So this is an important region that's really far downstream that is important to consider there. Of course, in the upstream region, it's important to consider the promoter.
But why not taking a step back and just look at it completely unbiased, not knowing anything about genes or which areas are regulatory or not, and then just because probably when insights progress, there are probably other levels of regulation that we don't know yet now, and that may actually be relevant also clinically. Why not just looking at genetic variation per se, cover that well, saturate the genome with markers that basically cover all your genetic variation as far as that is possible. Of course, there's also always some small parts that are not an LD with anything, but then look at it that way basically and skip the whole gene ID and just look at which areas are actually.
That's clear. ORAEST, that means, Mr. Candidate, that the time appointed for the defense of your thesis has passed, and the degree committee will now withdraw to the online delivery room to discuss the quality of your thesis and your defense. And I ask you and your company to stay online and await the results of our deliberations and our return. We'll be back. Are we already in the other room?
You can go to the other room by yourself.
Normally it goes very well.
By the leave button, leave room.
My leave button has a choice. Only it's now red and not blue.
Did
it
you like it
only
this way?
leave. And if I push that, then I can cancel it, but I don't have a choice. There's
Don't
no
have
blue
a
button.
choice. Okay, I'll check
No
it.
blue button.
Same for me, yeah.
I'm going to check it. On the bottom of your screen, there is breakout rooms. There are 4 dots in the symbol. If you click on that, you should choose.
I don't see it. On my iPad, I can choose perhaps for.
Oh, you have an iPad. 1 second.
Yeah. And in the beginning, it was leave was blue, and then I could make a choice between 2 rooms.
Yeah. No, it's not.
No, all cams are.
Otherwise, we leave you alone.
You let me have a look. The iPad. Linksboven heeft u de optie voor de iPad-gebruiker. 4 zwarte vierkantjes met de tekst Join Breakout Room.
It's easy if you are familiar.
Daar dient u op te klikken.
Ja, join breakout. I see
You
it
have to
now.
choose in the more option.
Then they are all gone. You had
Okay, you are all
41
here.
viewers
I leave
on
you for the
YouTube,
deliberation.
so you were not
Have a
alone.
nice deliberation.
We'll meet again somewhere between in 10 minutes, I think. Yes? See you later.
We managed it quite well, thanks to the support. Hey. Ja.
Wacht even. Zo. Dat is dus knudden, hè?
Ja, dan moest er de pauze inzetten, yes.
Ja, maar dan ken ik dus niks meer.
Nee.
Ik kan dus nu niet controleren of dat
Ik zal het nog
pauze
een keer
is.
zeggen. Het is een support stream. Wat ik gedacht, oh, je hebt OBS. Ik heb een pauze die aanzet.
Ja, maar dat is dus kloten. Dat is...
Daarom deze methode is gewoon shit. Maar we kregen een compliment van de prorector.
We are in the sharing.
So where are we? You're all there?
The candidate is having a cup of tea, coffee, or whatever at this very moment. You are very early.
Yeah, but with only 5 opponents, and
I understand. I understand.
Yeah.
Our candidate is arriving.
Good. And the candidate can hear us again. Thank you. Mr. Matern.
1 second, please.
Yeah.
Go ahead.
Yes.
Yeah, Mr. Matern, you hear us? Yes. The degree committee here present online has assessed the quality of your thesis and your defense. In view of its positive verdict and taken into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Tilanes is authorized to confer upon you this academic distinction in accordance with the Dutch university custom. I invite your supervisor to now take the floor.
Thank you, Mr. Pro-Rector. By the authority vested in us by law and in conformity with the decision of the committee here present online, I hereby confer upon you, Benedict Mark Matern, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, you will soon receive the degree certificate signed by the rector, the secretary, and the other members of the committee and affixed with the official seal of the university. As proof of your achievement, you receive this letter of confirmation signed by the rector, shown by the beadle. So, dear Dr. Matern, dear Ben, congratulations to you and your family and partner for having this, that you reached this stage of the doctor degree and with all the achievements you have done in my lab during this PhD thesis. And Dr. Groeneweg will say the laudation to you. So I give the word to Matthijs Groeneweg.
Hi, esteemed Dr. Matern, dear Ben. It is also my great pleasure to congratulate you on obtaining your PhD title. A great achievement that you managed to do within the allotted 4 years, which is very impressive. You were the first applicant that I had an interview with that was fully online without any live contact. Seeing as how the things are at the moment, that turned out to be, well, prophetic. All of us were impressed with the interview and, combined with the recommendation you got from Martin Myers at Be The Match, we decided to offer you the PhD position. We actually met each other for the first time, not in Maastricht, but at one of the Be The Match hackathons. I can't quite remember which one though, because there have been so many. And that was a few weeks before you came over. Shortly after, you came to Maastricht, and for the first few days you stayed at a youth hostel under the Kennedy Bridge. That was not an ideal situation. However, you soon found an apartment, and together we organized some furniture for you and got you moving in there. The project you were initially hired for was to develop a new program to analyze and annotate HLA alleles with data that came from a new technology called Oxford nanopore sequencing. As these things go, we greatly underestimated the effort that was needed to produce such a program, especially because you had to do it all on your own. However, you persevered, and although HLA Prospector, as the software suite is called, never got to the point where it was used routinely in diagnostics, it did and still does do a good job in specialized analysis for research and development, and has been used in quite some publications since. Another part of the Prospector suite is the submission tool Saddlebags to submit data into the IPD IMGT HLA database. This program is now used in several laboratories worldwide, and you published your first paper on it. As a final part of the utility section of your book, you published a paper on the use of Oxford nanopore sequencing in diagnostics, and that has been discussed today quite extensively. You attended several conferences and meetings, like the EFI and ASHI conferences and several hackathons. And apart from the scientific sessions, it enabled you to meet with your old colleagues from Be The Match and have fun at the parties and other festivities. I still remember the party at the Baltimore Aquarium. That was very special. At these meetings, you gave both oral and poster presentations, and you managed to get the best poster reward at the 2019 EFI meeting. You also joined the hackathons organized by the Be The Match, and these put you in contact with the wider HLA bioinformatics community. So hopefully these meetings will continue to bring bioinformaticians from everywhere together. While in the Netherlands, it was clear that you missed your family from time to time, and understandably so. From your stories, they sound like a great group of people, and even I get a Christmas card every Christmas from them. I'm still surprised by that. Luckily, you were able to combine some of the US meetings with a visit to your family, and hopefully, combined with the yearly Christmas visit, that eased the stay at the other side of the Atlantic Ocean somewhat. Soon after your arrival in Maastricht, you met new friends through the International Student Network, and also some of our colleagues became very good personal friends to you. Especially board games, movies, and escape rooms were, and probably still are, done a lot. Actually, in the end, I think the board games put you in contact with Yvonne. Meanwhile, your work progressed from programming basic analysis software to more scientific subjects. You continued to work on the HLA-DRA, the DR13 haplotype, and together with Diane Santis and Ling Truong, you looked into the HLA-DPA-DPB haplotypes. To help the Perth lab with the installation of saddlebags and to look into the DPA-DPB paper, you spent a few weeks in Australia twice, emphasizing the importance of international cooperation. All this resulted in some very nice papers, some of which you presented today. As a continuation of shaping international cooperation, we have now moved to Utrecht with Yvonne to start the next phase of your life. Here, you are going to help the HLA community get ready for the next bioinformatics-focused International HLA workshop in 2022. And you're going to do this at the Lab of Air Experience. Unfortunately, the Corona crisis interfered with the original planning of the thesis defense, but in the end, you found that does not matter anymore, as you finally did get your PhD today. Again, also on behalf of Marcel and the team in Maastricht, congratulations on a job very well done to you, your family, and all your loved ones. And with that, I would like to return the word to the pro-rector.
Thank you. Dear Dr. Matern, also on behalf of the Board of Deans, I congratulate you with the honor you have acquired. And it's a strange situation, in fact, but I hope and trust that you have somewhere a party going on when it's all allowed. And well, this is some real great thing in your life to experience to get a PhD. So congratulations again. And also congratulations for your supervisors, which we normal congratulate during the reception, but I can do it now here. Congratulations for the whole team. And I would like to thank all participants in this academic participants, also the Beadle, of course, who has came in right on time with the Ora Est, and also for the technical support of Luc Peters and Ger Verwunnik, because they really made things go very well and very smooth. Having said that, I now declare this ceremony to be ended. Thank you very much.
Thank you.
Thank you very much. I will stop the recordings. I will stop the live stream. If you would like to have a kind of online reception, you are free.
Is there online alcohol?
Yeah.
Under the table.
Yeah. If only.